Our next video series is going to focus on one of the most widely used statistical techniques, which is analysis of variance, or as it's often abbreviated, taking the first two letters of the first word, the first letter of the second word, and the first two letters of the third word, ANOVA. As an example that we're going to work towards, I wanted to show you a data set I've been working with recently. It comes from Dr. Timon's lab at University of the Pacific. Now, Dr. Timon's lab studies resistance in mosquito populations. We all know it's important to control for mosquito population growth in certain areas because mosquitoes are natural vectors of a lot of diseases. So one of the theories about resistance in mosquito populations is that some of them develop resistance by increasing the levels at which they express certain enzymes, one of which is oxidase. So in one of the projects from Dr. Timon's lab, they collected mosquito samples from a bunch of different local areas here, Amoruso, Brandon Island, etc. And for each mosquito, they performed some biological bioassays, enzyme assays that I don't completely understand, and come up with a number, an expression level for oxidase. And what you see before you is a box plot of this data by location. So the natural question to ask with this is, do mosquitoes in some locations tend to express oxidase at higher levels than mosquitoes in others? And I should also note that this is a log plot because, as you can see, the expression levels take place across almost an order of magnitude there. So this is a very different scenario than what we worked with in regression, right? And I wanted to give you a brief outline of how we're going to be laying out this series. This video you're watching right now is actually going to be part one, called a new test. We're then going to continue on to part two, which is LRT strikes back. We're going to come back to the likelihood ratio test and use that to develop a actual test statistic and rejection region for an ANOVA test. And then finally, in part three, we'll see the return of ANOVA. We're going to come back, see how to implement all this in R, and hopefully that will all end well. Now, the first example is a little complicated because of the large number of locations and also the large sample sizes per location. So I wanted to start off by discussing a toy example, which will hopefully help us develop a little bit of intuition and just kind of some visualizations to use as you tackle ANOVA problems. So in our toy example, I'm going to pretend we are researchers looking at a group of 9 subjects. And what we're interested in can be up to you. I'll leave that up to your imagination. But we're going to split those researchers into 3 treatment groups, Group 1, Group 2, and Group 3. And in our scenario, we're just going to use equal sample sizes in each group. So throughout ANOVA, we're going to typically use the parameter little k as a representation of the number of groups. So in our toy example, k will be 3, but in the mosquito example, k will be larger. Now, let's say that we split them into these 3 treatment groups and we take measurements on each of our subjects, and we end up with data shown in this table here. So those subjects in group 1, there was 3 of them, right? They had scores of 4, 5, and 6, giving their group a mean of 5, right? Group 2 had scores of 3, 4, and 5, giving their group a mean of 4, and group 3 had 3 scores, giving their group a mean of 7. So looking at this data, it appears that group 3 had the highest sample mean scores for this assessment, whatever it was. However, of course, as statisticians, we want to know, did the groups differ significantly in performance? So are these differences in sample means representative of a difference in actual population means? Okay, everything clear so far? Hopefully. If not, pause, do what you need to do, get a cup of coffee, get some water, and we'll move on. Before we lay out the null and alternative hypotheses, I wanted to introduce a little bit of notation and get you comfortable with the double subscript notation that's common in a lot of ANOVA examples. So here's that data from the previous slide again. In ANOVA, we often use double subscripts because we have 2 sort of things going on, right? We have the group numbers, and then we have the observation numbers within those groups. So we're going to use the notation Yij to represent the value of sample I from group Z. So for example, looking at our table here, right, if I focus on this entry, that would be sample 2 from group 1, so that means Y21 would be 5 in our case. And of course, as usual, if I'm thinking of these samples as random variables, so thinking about their distributions prior to doing the experiment, I would use Y21 or something like that to represent the value to come, right? Now, there's also these values at the bottom of the rows, which are the sample means for each group. And one of the common ways of denoting sample means within ANOVA is to use this dot notation. So notice that for this first sample mean, for example, I am averaging all the observations in column 1 of our data frame. So since I'm averaging over the row index, the first index I here, I'm going to put a dot in that index and then leave j in the second. Well, j would be 1 if I'm doing group 1. Okay. So the group sample means are defined by the formulas 1 over nj times the sum j equals 1 to nj, excuse me, that should be an I there, right? Hope you caught that before I did, of the yij's, right? So we're adding up all the observations per group and averaging them out. Sometimes, because that gets a little tedious, we might also just use the notation yj bar in certain situations. Now, the grand mean of all the observations is what you get if you add up across everything in the data set. And you can see here that I messed up my indices again here. That first one should be j and the second I. So the grand mean is what we get when we average all the observations. And it's actually a double sum, but oftentimes you'll see we're just going to simplify this by saying a sum over all ij of yij, and just understanding that when I have two indices here, it's going to be a double summation. So with this notation in hand, we can actually make some comparisons with the simple linear model. So in the simple linear model, what we were doing was trying to look for evidence of a linear relationship between a numeric response and a numeric predictor. And that led us to describe our model, which was that our response variable, right, y was the response, is related linearly to the predictor plus some noise, okay, where those noise terms we modeled as IID normal random variables with a constant standard deviation sigma. The ANOVA framework is slightly different in the sense that now we have a numeric response, but we have a categorical predictor, right? A categorical predictor that could be the group number in our first experiment or the location in our mosquito experiment. So the model now, the mathematical model, is we're trying to see if the yij's are a group mean, so this would be a population group mean here, plus some IID noise terms, where those noise terms, again, we're going to model as normal random variables with a constant standard deviation sigma. So the null and alternative, then, you should be able to guess what they are, right? Under the null hypothesis, all of our samples, there is no difference between the groups. So all of the samples come from the same population. So H naught is going to state that all the groups have the same population means. In other words, if I look at the mu1, the mu2, as many means as I have, as many groups as I have, right, they're all equal to some parameter mu. On the other side of that, under H1, we're trying to really assess to see if there's actually evidence that they're different group population means. So for example, in our toy one, right, if H naught was true, then our treatments didn't make any effect and all the subjects essentially had the same average scores on a population level. Under H1, then our treatment did make an effect. One of the groups had a higher or lower mean than the others. To visualize this, as we move on, I want you to think about the fact that under the null hypothesis, whenever we're doing an ANOVA, the null hypothesis is basically stating that all of the results come from the same normal PDF, whose mean then, right, we are estimating with what we called the grand mean of all the observations. In our toy data set example, that was 5 1/3. Right? On the other side, under the alternative is true, then the results potentially came from different normal distributions, right? Where each of the normal distributions now, for example, in the blue group, this would be centered at the sample mean for that group, right? So as we move forward through the other videos, we're going to get a little bit technical. We're going to do some algebra, walk through the garden of algebra many times, as my friend Parker Sperlin likes to say. And I want you to keep these pictures in mind as we do so, that really at the end of the day, what we're testing is to see if we have one normal distribution or many different normal distributions involved with our data.